Hello, everybody. How's things? How's it going? Hope everyone is faring well with the kind of lockdown orders. Hopefully, things seem to be soon to opening up, kind of, sort of. Right, a lot of states are finally starting to, open up to certain businesses and things like that. So hopefully, not too far off from that here in PA as well. I was going to say the kind of the last part of the special senses that we are going to be getting into is talking about our sense of hearing as well as equilibrium. Right? Now, that's both of those things are kind of transmitted via the inner ear. Right? We think about the cranial nerve that's responsible for this. Last class, right, we had mentioned and talked about something called the vestibulocochlear nerve. That was cranial nerve 8. Right, that functioned to transmit our sense of hearing, as well as those impulses from the cochlea to help with our, again, what we call equilibrium. Essentially, think of it this way, if you were to close your eyes, right, go ahead and close your eyes, tilt your head down. Oh, okay, how did you know your head was down? Right, you could think, oh, well, you thought about turning your, looking down or tilting your head down, but how is it that you actually know? How is it that your brain actually knows that you're in that position? Right? One of the ways is because of the impulses being sent to it from the inner ear. Right? So, let's see, before we get into that, we're going to just be looking at some of the different structures, right, associated with our sense of hearing. Right? So, different parts. Different parts, right? I point this one out first because it's kind of the delineation, kind of the marker, right? The divider, right? Anything this way is a part of what's called the external ear, right? So, like, all of this is a part of the external ear, right? All of this stuff, right, that I just circled, that's a part of the external ear. Alright. So out here, right, we see something called the auricle, or something called the pinna. Your choice, either one's correct. Right, that's the flappy part. Right, that's the thing that you can fold on the side of your head. The whole purpose, the whole idea. Sound waves, right? We make sound. Right? Those sound waves, right, as they travel through the air, we want to direct them into the actual ear. We want to direct them into what's called the external auditory canal. Right? This opening that you insert a Q-tip into, or at least you're not supposed to, but I'm telling you, it's fate. They fit perfectly. Right? It's like a match made in heaven, but if you look on the box, it says, oh, don't insert into your ear canal. Ooh, man, it feels good. Anyways, right, when we think about the external auditory canal, right, that's the structure all in through here. Again, as I mentioned, the sound waves get directed into the external auditory canal, and they come into contact with this structure right here. That's something called the tympanic membrane. The tympanic membrane. All right, that's important. We're going to talk about that in a second. But in the external auditory canal, right, something we see, something called cerumen. Right, that's just earwax. Right, there are glands called ceruminous glands. Yeah, they're just glands that produce the earwax. Right, so again, this would be cerumen right here. All right. So that was from here that way. Right, it's all external ear. Cool. Now we're going to get into a part of the middle ear. That's all a part of what's called the middle ear. The middle ear. So what happens, right, in order for us to be able to hear? Sound comes in, it's directed into the external auditory canal via the auricle or the pinna, right, whatever you want to call it. It comes into contact with the tympanic membrane. Right? If you have a drum, right, or even if you've got a thing of Tupperware, right? Put the lid on the Tupperware, and then hit the lid. It makes like a thud, right? But yeah, what happens is you are actually hitting that drum, right? Or you're hitting the top of the Tupperware lid, and it's making a sound. Yeah, you're actually using force, physical force, to generate a sound. The tympanic membrane itself, right? That's what helps transmit that, kind of turn that sound wave into a mechanical type of force. Right? Think of it this way. If you've ever been at a party or been anywhere where you've had a loudspeaker on, right? Let's say the bass was real deep and heavy. If you had something to drink, right, whatever it happened to be, when the bass would hit, right, that liquid in your drink would kind of vibrate a little bit. Right? That's exactly what happens in the inner ear. Right? When that fluid gets ripples in it, there's little tiny cells, right, in the inner ear which then kind of decode that message, right, depending on what the ripples are. Right? But we'll get to that in a minute. Cool. So sound waves enter into external auditory canal, hit the tympanic membrane. The tympanic membrane vibrates just like a drum head. Now there's 3 ear bones, or what are called ossicles. There's the malleus, here. There's the incus, here. And there's the stapes, which is here. And you'll kind of see the malleus is attached to the tympanic membrane. The incus is attached to the malleus. The stapes is attached to the incus. Right, so what happens when this occurs is, think of it. Because of the fact the malleus is attached to the tympanic membrane, when the tympanic membrane vibrates, that causes the malleus to vibrate. Right, so the malleus vibrates. Cool. But when the malleus vibrates, that in turn causes the incus to vibrate. When the incus vibrates, because of the fact the stapes is attached to it, it causes the stapes to attach to it. Now, part of why this is, right, look at the size of the tympanic membrane. Right, look at the size of the malleus, the size of the incus. Look at the size of the stapes. Stapes is tiny. This wee little tiny bone. If you've ever ridden a bike, and at this point I hope you have, and if you haven't, whatever. If you haven't, that's fine. Right, but think about it. When you ride a bike, just think about the cogs, or just think about the gears on the bike. Right, think about the sprockets, the gears that the chain is attached to. Now think about which one is. Right, think about the big giant gear on the pedals and the small little gear on the very back wheel. Right, and just think about it. Which one has to, if you make one revolution, if you think about how fast that big gear is turning, that you're turning with the pedals, right? Think about how fast that small little gear is turning on the back pedal. Right? Think about it. Just kind of just normal how gears work. Right? If you have this big gear which is spinning, well, if that's attached to a much smaller gear, that smaller gear has to rotate that much more quickly. Same idea applies here. It's not that the stapes that it rotates, but the fact that the sound waves come into contact with the tympanic membrane, as well as the malleus, right? But they are massive, especially when compared to the size of the stapes. So because of that, that force is magnified significantly, right? And the stapes vibrates much more quickly than does the tympanic membrane, right? Just like that big, large gear that you're turning with the pedals of the bike, that's rotating much more slowly than the corresponding small gear on the back wheel, right? The smaller it is, the faster the rotation, or here, because of how small the stapes is, the faster it vibrates. And the faster it vibrates means it has a much greater amount of, or a much greater ability to kind of vibrate the fluid. Remember those ripples I talked about a few minutes ago? If you have a drink next to a boom, oh my God, I almost said boombox when I'm from the 80s, right? If you have that drink next to a speaker with a lot of bass, right? That stapes, because of the fact that it vibrates so quickly, creates a huge amount of ripples in the fluid. Right? We'll talk about the fluid and the effect that has in a little while when we dive a little bit deeper into the inner ear. One of the other structures that we see, right here, right? Something called the auditory tube. Right? It's also called the eustachian tube or the pharyngotympanic tube. Choice is yours, whichever one you want to call it. But what that does is it regulates the regulates the pressure of the middle ear to that of atmospheric pressure. Right? Remember, this, right, ignore this picture, just ignore this picture isn't here. Right? This will always be atmosphere. Right? Just think of all the atmosphere that's around you. Right? So the pressure on this side of the tympanic membrane matches that of atmospheric pressure. Right? Make sense? Well, in order for any drum head to work appropriately, or even for the tympanic membrane to function appropriately, the pressures have to be equalized on both sides. So the pressure on within the middle ear has to be the same as that of atmospheric pressure. This is how the auditory tube works. Right? You might think that's kind of weird. Well, yeah. If you just open your mouth, just held it open, imagine if I could shrink myself down small enough. Right? I could jump inside of your mouth, crawl up your tongue, crawl the back of your throat, and I could actually crawl into this tube. I could crawl right up in here. Right? So air, right? If I were small enough, I could do that. So that means air absolutely could do that. Right? This is why, again, when people have ear infections, or when we go up in elevation, right, up in an airplane, or living where we live, right, with the mountains and the altitude changes, if you're traveling on 81 or something like that, your ears kind of crack and pop at certain parts of the trip. Why does that happen? That happens because as you go up in elevation, or as you go down in elevation, that changes the atmospheric pressure, which in turn, now we have to be able to equalize the pressure on both sides of the middle ear. Right, so that's why things like chewing gum, kind of, your ears start to crackle and pop. So things like drinking, chewing gum, those things can help to relax some of the muscles of the throat, and that can help air to get up into and through the auditory tube and up into the middle ear to equalize the pressure on both sides. Make sense? All right. A couple other things, a couple other structures that we see here. This thing right here, something called the round window. Alright. Think of it this way. We haven't covered the inner ear yet, right? But we've covered how those sound waves, right, that vibration gets into the inner ear. Right? It goes from the tympanic membrane to the malleus to the incus to the stapes. Then from the stapes, it gets into the inner ear. Cool. There's a window, a little opening. If I could pull off the stapes, if I pulled it off, there'd be a little opening. There's something called the oval window. But that's the opening into the inner ear. Cool. So as that vibration enters into the inner ear, as it kind of swirls around, right, all that vibration, all that pressure is building up in the inner ear. There has to be some sort of way to release that pressure. And there is. Right? Think of the round window as being like the pressure relief valve, if you want to think of it like that. Right? In order for that vibration to occur in the fluid, there has to be some sort of way to alleviate the pressure. If you can't, more and more pressure builds up and the fluid doesn't vibrate or kind of ripple like it's supposed to. Or think of it this way. Go grab hopefully you're not able to do this example. Grab a bottle of soda. Right? It could be seltzer water, which is, you know, seltzer water or tonic or anything like that. Soda, I'm opting for soda. I hate soda. Man, tonic is blah. Anyways, take it and vibrate it. Or shake it up. Right? Take that bottle of soda and just shake it up. What happens to it? Pressure increases and the bottle gets super stiff. Very, very hard because the pressure inside of it has grown so much. Yeah, the pressure can't escape. It just continuously builds inside of the bottle. The same thing would happen in the inner ear if we didn't have the presence of the round window. Right? If that soda bottle is super hard, what happens as soon as you take the top off? Well, the pressure gets released, and the bottle softens. Right? Cool. Cool. So here, again, just another kind of up close and personal of the middle ear. Right? We can see the malleus, right, the incus, the stapes, right? We saw all these before. One thing that we didn't see in the previous slide is this muscle right here. Something called tensor tympani. That's the tensor tympani muscle. That's a muscle which helps to kind of dampen some of the sounds or noises that we hear. Right? Think of it, if we're able to, if a loud book, if you're at the firing range, you know, a gunshot, any kind of loud thing like that, you want to be able to dampen how much you're actually hearing, right? If somebody screams into your ear, what happens afterwards? Oh, your ear just begins to ring. Well, yeah, why is your ear ringing like that? It's ringing like that because of the fact that your tympanic membrane vibrated so quickly that it's caused your malleus, your incus, and your stapes to vibrate at huge levels. So it's just continuously beginning to ring. Right? So what the tensor tympani muscle does is it helps to pull malleus away. Right? Well, actually, the tensor tympani, I think it's stapes, sorry. Tensor tympani actually comes around, and when it happens, it actually pulls taut the tympanic membrane. So when that happens, right, if the tympanic membrane tightens up, or think of a guitar string, take a guitar string and just tighten the heck out of it, and then pluck that guitar string. Doesn't make a whole lot of sound because of how tight it is. Right, sure, the pitch will go higher and higher, but eventually it won't really make much of a sound because it's far too tight. Right, same thing with the tensor tympani. As it tightens up, it vibrates less and less, and it generates less sound. People, the elderly, right, for instance, we talk about like hearing loss. Yes, there are different things which can cause hearing loss, right? Let's say those things outside of the vestibulocochlear nerve, outside of the brain. If we look at hearing loss, kind of just good old-fashioned, hey, Grandma and Grandpa, oh my God, I had to, say hi to Grandpa 16 times because he couldn't hear me, right? That sort of thing. What's happening, right? In that sort of event, just like anything else as we age, right, as with most things, it decreases in function. No big shocker, right? Just like skeletal function decreases, muscular function decreases, GI function decreases, I mean, you name it. All these functions decrease. Receptors become less sensitive. Neurons become less sensitive. Ligaments become a little bit more lax. Right? Connective tissue doesn't heal. It doesn't heal appropriately. Not that it heals all that readily, all that well, anyways. But what happens as we age? Right? When Grandma and Grandpa can't hear. essentially what's happening, right, from point A to point B. Right? Point A is, well, there is, right, right, point A, right, is the tympanic membrane vibrating, and then going from the tympanic membrane to the malleus, to the incus, to the stapes, right, the ability to transmit that vibration into the inner ear. Because remember how I mentioned, once it reaches the inner ear, the entire responsibility of the ossicles, right, of the malleus, of the incus, of the stapes, is to transmit that vibratory sense, that vibratory force, into the inner ear. So when it does that, it vibrates, it sends ripples through the fluid, just like, right, just like you would have in a drink sitting next to a speaker with lots of bass, right? Because as that fluid moves, right, as the fluid moves, that's what's going to allow your brain to be able to kind of decode, allow your brain to be able to, make some sort of sound from what it is that you heard. Right? And next, we're going to get into the inner ear. But later in life, right, 60s, 70s, 80s, right, we have a reduced ability. One, the tympanic membrane vibrates less effectively. Why? Because with any tissue in the body, it doesn't heal as effectively. When tissues are 60, 70, 80 years old, right, they don't repair, they don't synthesize tissue the same. Right? Just like the muscles don't function the same at the age of 70, nor does the tympanic membrane. Right? The ligaments, which anchor the malleus, which anchor the incus and the stapes, they become more lax, meaning they have a little bit more, they aren't quite as tight. Right? They're a little bit more loose. So therefore there's less stability. Right? There's a little bit more or less stability throughout the malleus and the incus and the stapes. So, that's part of what's going on, is they have that reduced ability to get that vibration into the inner ear. Right? Now, one of the things with, right, this sort of one of the ways that hearing loss. I lost my train of thought there for a minute. Typical, right, with grandma and grandpa. Right? There's differences in what they're able to hear, right? If you ever noticed, like young children, right, for instance, women in general, I say in general, right? The reason why I say that is because usually, young children, women generally have higher pitched voices. As we age, right, because of the decreased vibration in the membrane, in the ossicles, but also in the inner ear, right, those receptors which are able to detect that vibratory impulse and transmit that impulse back to the brain, because that reduces with age, right, in a minute we're going to talk about amplitude and pitch. Amplitude is how loud you're talking, right? Pitch is just that, the pitch. You sound more like, Alvin the Chipmunk, right? Very high pitched or very low pitched, someone with a very deep tone. And the reason why I mentioned, young children, right? Women in general, right, have a more difficult time being heard by the elderly or those that have hearing loss, especially presbycusis, is because they lack the ability to detect those higher pitched frequencies. And we're going to talk a little bit more about that in a couple of minutes. Right? But keep in mind, it's that they lack the ability to detect those high pitched frequencies because those high pitched frequencies deal with a higher higher frequency of ripples, right? Or those higher frequency of sound waves. Right, we'll talk about that in just a minute. Right. Here's when we finally get into the inner ear. Right, so right here, the round window. Right here, the oval window. Right, this is where the stapes attaches to. Right, so the stapes attaches to here, and then we're going to ignore this stuff for a second over here. This is where it transmits that sense of sound, those sound waves, that vibration frequency along the cochlea, in through here. Right. In a second, we're going to look at something called the tectorial membrane. Right. That's what's going to allow your body, your brain, to be able to hear what it is that you can actually hear. Right. Actually, you know what? I'm going to jump to that now, so we can look at that, and then we'll talk about the semicircular canals in a minute. Right. But something called the cochlea, as well as the cochlear duct. Right. This snail shell looking thing. Right. This is the part of the inner ear which is necessary for hearing. Right. When you look at the whole inner ear, right, the whole thing kind of looks like a snail. Right. Here's the head of the snail. Here's the shell. Right. The shell, the cochlea. That's the part which is associated with hearing. All right. All right. So here, vibration enters into the inner ear, specifically the cochlea. All over through here. There's a fluid within the cochlea called the perilymph. Right. It's called the perilymph. I'm just going to jump to this next slide for a second. You can see it. I'm sorry. Right here. In this chamber here, right, this is where that fluid is, right in through here. Now we can see this thing right here is attached to this fluid. That's a tectorial membrane. Excuse me. That's a tectorial membrane. And you can see all of these different neurons right through here. We're going to blow up a little bit. Perfect. Tectorial membrane we just looked at. Right here are all those little hair cells. These are what are going to be stimulated, and these are what are going to actually generate that action potential to be carried back to the brain, so that when it travels to the auditory center of the brain can then decode what signals is being sent it, and you can then hear. So, back to the whole hearing loss, difficulty in hearing, right? Even in the elderly, right? When we talk about presbycusis, we talk about kind of good old-fashioned typical hearing loss. There's even a reduction or a reduced ability of the tectorial membrane to vibrate, as well as the response of these different hair cells. So when they reduce, when their ability to vibrate reduces, their ability to vibrate is very typically seen in the elderly. Alright, so now the tie-in to the pitch of the voice. Okay. Let's draw a chart, or a graph, excuse me. This is for, let's say, pitch. So, like, is your voice like high and squeaky, or is it kind of low and whatever, low and raspy? Right? The higher pitch, right? For instance, it doesn't matter. I'm not concerned about how high they are. Right? See how these are incredibly close together? That's someone who has an incredibly high-pitched voice. Let's compare that to somebody who has just a, right, let's say high. Let's say someone who's normal. Might look something like that. You can see the waves are nowhere near as close together. Someone has low-pitched voice, right? They're kind of spaced out like this. All right. So, if we think about high frequency, high pitched voices, look how close together these waves are. That means this tectorial membrane and these hair cells have to, right, that they are being vibrated unbelievably quickly because of the frequency. they don't function very well to begin with. Right? So their ability to send impulses that quickly is pretty kaput, meaning they don't have the ability to do that. Hence, so therefore, if that's what's being sent to the inner ear, right, they lack the ability, one, to transmit the frequency, to transmit the vibratory force from the middle ear, but then two, at the level of the hair cells, they have an inability or a reduced ability to actually convert that mechanical force into some sort of stimuli, right? Because they can't vibrate quickly. The person's 70, 80 years of age. These cells don't have the health. They don't have the ability to do that any longer. So that's why I mentioned if you speak with a lower pitch of a voice, right? Something like this. It's not that like, oh, I have this patient who's the biggest jerk. They just keep ignoring me all the time. But if I speak with a really deep, low, soothing voice, they can finally hear me. No, it physiologically makes sense, right? You want to speak loudly, but you want to speak with a lower tone, a lower pitch, right? Because if the wavelengths are spaced out like this, that would require the these hair cells to transmit that sense much less frequency, right? Or frequently, excuse me. Right, the tectorial membrane would vibrate that much more slowly. Right, because here, right, this is looking at a drink, and you've got the bass cranked up to the max. The bass hits, and you just see like 1,000 ripples in your drink. Right, that's high frequency. If you turn the bass down a lot, the bass hits one time, and if you look at your drink and there's just a couple of ripples in the drink, that's low frequency. Elderly have a much, much, much better time at hearing that low tone, low frequency, low pitch. Right. All right. That's different than something called amplitude. That's the how loud am I speaking. This has to do with the height. Right? So remember how tall they were before? Right? Someone who was speaking quietly, someone who's speaking normally, and someone who's shouting. Right? You can see the height of these is very different. Right? Someone who's shouting. Right? The amplitude is dictated by the height of the wavelength. The frequency, or how the pitch, is dictated by how close together they are. Does that make sense? So that's why somebody who's hard of hearing, you want to speak lowly, or excuse me, you want to speak with a low tone, a deep voice, but very loud. Right? Oftentimes when we speak loud, our voice elevates in pitch, which is the exact opposite of what we want to do. That makes sense? Alright, cool. Cool. So, if we back up for a second. So, the structures here, right, I wanted to finish that thought before I started identifying these other ones here. But the cochlear duct, this here, right, this is what we had gotten into. Right, this is what we were actually looking at when we looked right here. That's what we're talking about. And you can see it's kind of located in between the perilymph. Right? So these fluid-filled chambers here, that fluid is the perilymph. Fluid is the perilymph right here. Right. So we can see how within the cochlear duct is something called the spiral organ. Or essentially, this whole thing right here. Right? That's the spiral organ. That is the actual. Think of that as being like that is the actual unit which is stimulated in order for you to be able to hear something. Right? Whatever it is. Kids screaming, someone yelling at you, whatever it happens to be. Right? It's the organ of Corti, or the spiral organ, which achieves that. That's right here. right here. Right, we see how, remember cranial nerve 8 is called the vestibulocochlear nerve? Right, vestibulocochlear. Yeah, the vestibule is here. The cochlea is here. So we can see there's two different branches, right? See what kind of branches off. Right, there's a branch here. There's a branch here. This here is the cochlear branch. Right? The cochlear branch is what actually collects and sends that, right, that what you're hearing into the brain. The vestibular portion, the vestibular branch of the vestibulocochlear nerve, that deals with the vestibule. The vestibule, this is involved with your ability to kind of balance, your equilibrium, your ability to know if your body is moving or stopping, or if you're looking up or down, or if your head's rotated, and that sort of thing. Right? Talk about that in a second. So, now, as I wanted to kind of finish with our sense of hearing, right? Now I'm going to look at, back up, and then we're going to take a look at the part of the inner ear which is associated with your, right, kind of balance, equilibrium. Right, that's the head of the snail shell, if you want to call it that. Right, the head of the snail shell. Right, this whole part here. Right, this is what we're going to be talking about. Right, this whole part in its entirety. This is what deals with, again, your balance and equilibrium. Different pieces of it play a different role, right? For instance, semicircular canals, right, kind of give you an idea as far as, you know, think of like 3D orientation, kind of how you are moving from if your head's tilted side to side, right? The utricle, the saccule, they deal more with kind of starting and stopping motions, right? If you're stopping, right, how is it your body actually knows you stopped? That sort of thing. We'll take a look at those in just a minute. But here we're just going to look at where they act. You know, where are they? Right? Where are these things? What stands out the kind of the most? Are these big, kind of half circle things? I guess full circle. Right? These are the semicircular canals. You can see 1, 2, and then 3. What's called the ampulla, that's essentially just kind of like the beginning. Right, see the ampulla here, here, here. That's kind of just the beginning of the semicircular canal and where it actually attaches itself to the vestibule. But we can see, excuse me, the vestibule, and on this image it's a little bit difficult to see. I'm going to point it out here, and there's another image here in a minute. We're going to see it again as well. But the vestibule, right, this part right here, right, that's the vestibule. Just beneath that is something called the saccule, where the saccule is essentially between the vestibule and the cochlea. Right? And then above that, again, something called the utricle. Right, so think of utricle, U up, right, up, utricle. Right, in case that helps you. Right, sorry, backwards. So here, just another image of, right, the vestibular nerve, right, the vestibular branch or portion of vestibulocochlear nerve right here. All right, now let's fast forward. So here, right, this is when we talk about how our body knows, is my head down, is my head up, is my head side to side, that sort of thing. One of the ways that we are able to know that is because of what we're looking at here. This is inside of the utricle and the saccule. So, if you've ever been on a boat, or it doesn't matter, been on a dock, seen a movie where they've been on a boat, you know, looking into the water. If you kind of look into the, when you kind of look into the water, you can see that the seaweed, and depending on the current, depending on whether the water is moving one way, the seaweed bends one direction, whether the water is moving another direction, the seaweed may bend in a different direction. Okay? That same thing kind of happens inside of the inner ear, specifically the utricle and the saccule. Right? As our head bends from one side to the other, or flexes forward, or extends upwards, right? That's going to change and bend these different cells. Right? So these little crystals, right, we're continuing that thought. These little otoliths here, they're called otoliths. They're like little crystals. In the cases of like vertigo, for instance, this is something that might be going on, right? Where somebody has some sort of, right, like otoliths become kind of stuck, or have an inability to move like they're supposed to, right? The reason why that's a problem, right? Some people might develop vertigo from inner ear problems, to infections, things like that. Vertigo is more or less kind of when your inner ear doesn't match with what other parts of the body are telling the brain what its orientation is. Right? So for instance, if your eyes are open, all right, looking straight ahead, I'm standing up. Right? So because of that, these otoliths are oriented in a certain way, that these hair bundles are oriented in a certain way, that, okay, we all are in agreement my head is upright. If I then bend my head to the left, so as to touch my left ear to my left shoulder, my eyes almost stack on top of each other. Right? And even if I were to turn off my inner ear, I would know my head's turned to the side because of the visual information. The way that it's coming into the brain, my brain would know, our head's turned to the side. Alright, now we turn on the inner ear, and okay, that matches. The otoliths, the hair cells, they bend in a particular fashion, and our brain knows, my head's turned to the side. Awesome. With vertigo, that's where the pieces of information don't match up. Right? My eyes, for instance, turn my head to the left, while my eyes are stacked, right, essentially kind of stacked one on top of the other. That visual information enters into my brain. Right? Now, we're ignoring even the joint receptors and stretch receptors. They're all sending stuff to the brain as well. We're just ignoring those for a few minutes. But that's all sending information into the brain, and the brain says, our head's turned to the side. Awesome. If that doesn't match up with what the inner ear is sending, because the otoliths are stuck, they are seized, there's some sort of inner ear infection, tumor, whatever it happens to be, our brain gets confused, and our brain's like, well, I don't know what's correct. Is my head to the side, or is my head upright? And that's where we just start to spin, because the brain doesn't know what's up and what's not. Right? Or what is up and what is not, what is down, if that makes sense. Right? Okay. So as I mentioned, right, as you move your head, right, these crystals, these hair cells, they move. Right? So as for instance, as the head goes down, we can see these hair cells bend along with it. Cool. So if, for instance, you were to look up, right, these hair cells would bend backwards. Cool. That impulse, it gets sent into the utricle and saccule. That then gets sent into the brain, into many areas, cerebellum, temporal lobe, and a few other areas as well. Now, same idea, right? Same sense, right? Same concept. However, right, our head doesn't only move in forward and backward motions. Like for instance, right, the utricle and saccule, these guys here, right? This has a lot to do with how, right, whether our heads are bent forward or backwards, but really it has a lot to do with our sense of movement. Right? Think of it. Nowadays, and you got to have everybody buckled into the car. Everybody's got to be buckled into their car seat in whatever different way. Right? Growing up, people always make a joke. It's like, my gosh, back in the 70s, 80s, your parents talk about, they're like, hell, I was, I rode, my parents used to take the seat out of the car for long road trips. They would just bring an air mattress and lay in the air mattress in the van. Right? The reason why that is because if the van had to stop quickly, you're going to get thrown to the front of the car. Right? Same idea here. Right? If we, for instance, if I'm just moving forward, right? Ignore this guy. Ignore this thing here. Just ignore this. Crossing them out. We're ignoring that. If I'm just walking forward, and all of a sudden I come to a just stop. Right? Well, these ordinals are gravity or momentum is carrying them forward, and if I stop right here, they're going to keep going for a brief period. Right? They kind of stop here, and then they sling back to their normal position like they're supposed to. That's telling the brain, hey, we just stopped. We're not moving any longer. Right? Does that make sense? Right? And that's the thing. The same thing with if we're going faster and faster. Right? Depending on how fast we're going, if we're going super fast and all of a sudden we make a quick stop, well, depending on how far forward these things bend, that tells the body how fast of a stop we just made. Now you might think, well, what the hell does that matter? We already made the stop. As you are stopping, as our brain is, as we are stopping, as we're slowing down, I get it. If you videotaped it and put it on a 10,000 frame video, right? Or as it's occurring, it takes like a second for you to stop. But if you froze it down into thousands of frames of video, it would take a much longer period of time. And the reason why I say it like that is because even though how fast it's occurring, the reason why it occurs that quickly is because the muscles are causing it to occur that quickly. How do those muscles know what to do? Because they are the only way they know what to do is if they know how slowly the brain is slowing down to begin with. Right? Like if we know how fast the body is slowing down. Right? If the body is slowing down rapidly, then all of a sudden we've got to change and shift weight onto different legs, and we have to be able to balance ourselves. So once we go from running 20 miles an hour to within 4 steps come to a dead stop, we've now got to shift our weight, alter our center of gravity so we don't then topple over. The reason why we're able to do that is because of these otoliths and these hair cells within the inner ear. Right? So here is the same idea, but these deal more with the semicircular canal, right, as opposed to the vestibule in the utricle and saccule. Right? So this thing called the cupula or the cupula, however you want to call it, we see hair bundle, hair cell, supporting cell, very similar. Right, hair cell, right, very very similar to what we had seen previously. However, one of the things, right, that we're going to see, so for instance here, right, position of the cupula, with the head still in position, the head rotates. Yeah, when the head rotates, right, because of the fact, right, as our head rotates, well, does that change gravity? Right, so, right, let's back up. If our head rotates, inside the within the vestibule, inside the vestibule here. That's just kind of one location, right? And what I mean by one location, I mean if I'm going to talk about the semicircular canals, you get my point. These are big looping circles in multiple planes and orientations: the coronal plane, the sagittal plane, the transverse plane. Right? The vestibule, sagittal, utricle, they're not like that. Right? We can see. Again, we're talking, this is within the vestibule. We can see how, well, this responds to gravity. Okay, cool. Well, what happens if we don't bend our head forward? Or we don't bend it side to side? What happens if we just rotate it? Well, if we just rotated it, these wouldn't move at all. These would stay incredibly stationary. Right? Does that make sense? Right. And one thing that, again, the utricle and the saccule, they are parts of the vestibule. Right? So I keep using them interchangeably. Right? The vestibule is kind of the big picture. The utricle and the saccule, they make up. They're within the vestibule. Right? But this is why I kind of talked about the vestibule really is more involved with kind of, acceleration and deceleration, slowing down, speeding up, that sort of thing. Whereas the semicircular canals, because of the fact that they are, again, oriented in these multiple planes. Even if we only just rotate the head this way, rotate the head this way, we are still able to recognize that. Right? From an orderless standpoint, that we can't do. Right? But if we turn the head to the right or to the left, because of the way that the semicircular canals, because of the different planes in which they exist in, that still allows the cupula, or these hair cells within the semicircular canals, to bend. Therefore, we are still able to figure out how the head is moving. Is it rotating? Is it shifting? We're still able to figure out how it's actually moving. Right, does that make sense? Alright. Cool. So those were the 5 senses, right, in the previous presentation. Right, we covered taste, taste, vision, smelling, right? Hear, right, our sense of hearing, as well as kind of balance, equilibrium a little bit. There's about a half an hour left or so. I'm gonna the next video is going to be of kind of a little bit of a kind of review from what's been on, right, the last 3 or so PowerPoints, or the last three or so presentations. Right, as I mentioned, because of the way class has kind of gone, right, because of the fact we can't be in the classroom anymore, right, because we can't be conducting class the way we typically would have been. I've talked more about function than I generally would, right, and that's just since we don't have the models on hand to be able to kind of belabor the point or to look at the different connections. Right here, I'm just talking about the functions a little bit more so therefore we talk about the functions, and if you realize what the neuron does or what the hair cell does, that may help you then remember where it is and kind of what location it exists in. Right, so I will be going through the PowerPoints and getting all of the different images, right, and putting them on D2L. Right, that way you guys can see all of the images I will be utilizing for the exam, along with the structures that I'll be utilizing. Right, so we're looking for that on D2L. That should be up. That should be up on D2L already. So look for that. It'll be in the content section as well. It'll be in the content section. All right. So, anyways, I'm going to post a review here in a little while. So if you guys have any questions, please reach out. Right, and we will talk soon.